So good morning. It's a pleasure to be here. So I'm actually what's called a Komen scholar, which means that I've been fortunate enough to get funding from Komen to do my research, which is mostly on trying to develop new drugs for breast cancer now for several years. I actually don't remember how many years, but it's really helped allow me to do the stuff that I do. So I'm very appreciative from Komen, and I'm happy to talk about some of that today. So there's a number of very encouraging drugs being developed for metastatic disease right now, but I want to focus on one class of drug called immunotherapy, partly because it's probably one of the fastest moving fields right now, and the other is because it potentially has the ability to make a really pronounced impact, both in metastatic disease and then eventually, hopefully, in patients with earlier stage disease. So just as a show of hands, how many of you have heard of immunotherapies for cancer? Great. So it's actually, there's quite a lot of hype about it right now. In fact, if you drove in on the Pike today, you might have seen these billboards for Keytruda, which is a very opaque billboard, but because all it says is Keytruda. But Keytruda is one of these new immunotherapies, and so I think you know a drug or a class of drug has made it when it's on a billboard on the highway. So you've all heard of it, but I just wanted to kind of walk through a little bit about the basics of really what it is, because oftentimes it's a concept that it's very different than essentially all of the other types of drugs that we're developing right now. So I think when people think of the immune system, they think of the part of our body that fights infection, and clearly that's very important. So the immune system is first and foremost designed to kill bacteria, kill viruses, and get rid of an infection. But actually, what the immune system is really designed to do is kill anything in your body that's foreign. So anything that the immune system recognizes as not being supposed to be there, it's designed to get rid of. And so that's why, in fact, that the immune system is what's responsible for rejection of transplanted organs. So when a person gets a new kidney from another person, the immune system would recognize that as foreign and actually goes out and tries to kill it. And so anybody who has a transplanted organ has to be on what's called immunosuppressive drugs, essentially for the rest of their life, otherwise they would reject that organ. And that's, again, the effect, and that shows you the power of the immune system. I mean, my kidney is only slightly different than someone else's kidney, but the immune system is able to recognize the slight differences between my cells and someone else's cells. And along those same lines, the other thing that the immune system is designed to do is kill cancer cells. Because again, because of the mutations that happen in cancer, the immune system is able to recognize the differences between a normal breast cell, for example, and a cancer breast cell because of these slight differences. And that's been recognized for a long time by scientists, that the immune system is designed to kill cancer cells. But obviously there's something going wrong when a person develops a breast cancer. Clearly, the immune system is not doing what it's supposed to be doing and allowing that breast cancer to evolve. And that idea that, well, we just need to fix whatever that problem is, why the immune system isn't working, get the immune system to work, and it should then be able to get rid of the breast cancer. And so, that idea has been around for many years. In fact, when I was in college, this was on the cover of Newsweek, and it's a guy named Steve Rosenberg who was at the National Cancer Institute, and he had developed an immune therapy back then, and there was a lot of excitement that, hey, this was a way we were going to cure cancers because we were going to get this immune system to start doing its job. Unfortunately, back then, the way they were doing it was with drugs that basically stimulated the immune system very non-specifically. So people got very sick because their immune system would just go crazy. It only worked in a very small percentage, a few percent of people. And so that this approach kind of died out, but it did spur this idea that hey, if you are able to get the immune system to work, it can make a real impact on cancer. But because of the the flaws of this particular approach. Things were kind of dormant for many years. But that's all changed, and it all changed a few years ago. And now, as you have all heard, the immunotherapy is looks like it's a very powerful approach. And in fact, now it's back on the cover of magazines. It's been the cancer breakthrough of the year now for several years, and it really, is now here to stay. And the real breakthrough, so why do we go from a failed therapy in the 1980s to what clearly a successful therapy now, is all because of one clear breakthrough. And that is that the immune system has an off switch, actually an on switch and an off switch. And that kind of makes sense. When you have an infection, you want the immune system to turn on and kill that flu virus, but you don't want to be sick for the rest of, you don't want to be having the flu, the fever and muscle aches and feel lousy for the rest of your life. You need the immune system to turn back off. And of course, there is that. We know that happens. Once the flu virus is gone, the immune system goes back to a resting state, and you feel back to normal. So it was the discovery of how that system works that really led to this development of effective immune therapies. So I need to talk a little bit about the science here, and you'll have to pardon my very basic cartoons, but this is the way I think about this. So the immune system is composed of a number of different kinds of cells, and the kind of cell that's designed to kill cancer cells is called a T cell. And what happens is the T cell recognizes the cancer cell by something called the T cell receptor, and it binds to something on the tumor cells that it recognizes as different. And so the T cell recognizes the tumor cell. The T cell then becomes activated and basically sends toxic things towards the cancer cell, and the cancer cell dies. So all well and good. That's the way the system is supposed to work. But as I said, once the cancer cell is dead, the T cell is supposed to turn off. And the way it turns off is that once it's been activated, it expresses another protein called PD-1. And so that turns on, and then eventually, once the infection, or in this case the cancer cells, are all dead, that switch is supposed to be activated to turn off the T cell by other immune cells. The problem is that cancer cells turn out to be kind of smart. And so the cancer cells recognized that these T cells have this off switch, and so they made a protein called PD-L1, which is a protein that binds to PD-1 and essentially pushes that off switch. So once the once the tumor cells start making this off switch, they are able to turn off the T cell, and now the tumor cell lives. So once this was discovered, and actually was discovered by, at least in part, by a scientist at Dana-Farber, that there was this off switch on T cells, and that the tumor cells were using this as a way to evade the immune system, drugs were developed that block that interaction between PD-1 and PD-L1. And that's these antibodies that are on billboards, drugs like Keytruda. And so if you block that interaction, you prevent the tumor cell from turning off the T cell. T cells then is allowed to be activated again, and the tumor cell dies. So this was, a laboratory development, just like many laboratory developments, and this worked really well in mice. You could now cure lots of mice with tumors by turning off the ability of these tumor cells to inhibit the immune system. So looked encouraging, but again, lots of things look encouraging in mice. But this one turned out to be active in people as well. And so there was this back in 2012, so only now 4.5 years ago, the first study of one of these drugs that block PD-1 was published. And these are the data. And I'm sorry to show kind of really a graph. I know not everybody likes graphs, but this is worth seeing. So this is a graph of the size of people's tumors on this trial. And these were patients with metastatic melanoma. Those of you who don't know, metastatic melanoma is a very difficult tumor to treat. It really doesn't respond to chemotherapy, and really there are very few options for patients with metastatic melanoma prior to this. But so patients with metastatic melanoma were treated on this trial, and each of these lines is a single patient, and this is the size of their tumor. And you can see that most of the patient's tumors on this trial basically stayed stable, or they got smaller, and only a very few number actually continued to grow. So this doesn't happen in metastatic melanoma. There's never been a trial like this that showed data in metastatic melanoma like this, and this was the first trial of immune therapy. So this was really quite remarkable. And what's particularly useful or particularly impressive is you look at the scale here. And so these were weeks. Most of the time metastatic melanoma trials are, this axis is days. And so this is weeks, and you can see that a lot of these patients are out 2 years with complete stability of their cancer. And again, it just was never, that kind of data has never been seen before. So that was really groundbreaking. And it brought up this question as well, yes, the drugs might be working, but why are they working so long? We never see that in metastatic cancers, particularly melanoma. And it's probably because the immune system, a lot of our drugs, they target one part of a cell, of a cancer cell. We have drugs that block estrogen receptor. We have drugs that block HER2. And so if you're targeting one part of the cancer cell, it's a little easier for the cancer cell to change something so that change that one protein, the one target of the cancer cell, of the drug, and allow the cancer to now evade that treatment. And that's how drug resistance happens, and Nick is going to tell you a little bit more about that. But when the immune system isn't targeting just one thing, it's targeting everything about the tumor that's different. And so it's a lot harder, theoretically at least, for the cancer cell to evade lots of essentially it's lots of different drugs than it is to just to overcome a drug that's targeting one thing. So that's one issue, why these immune therapies seem to be working for so long. And the other is that one of the ways that cancer cells escape our treatment is they mutate. And that's how they keep changing to kind of get around our treatments. But as I said, the immune system is designed to find these changes. So as the cancer cell is changing, presumably it's developing more differences that the immune system can then recognize. Again, these are kind of theoretical ideas, but it's probably at least part of why these immune treatments are working for years as opposed to weeks or months. So that's melanoma. Really been completely groundbreaking and game-changing in melanoma. But we're a breast cancer group here, so let's talk about breast cancer. So the first treatments of immune therapy in breast cancer have focused on the kind of breast cancer called triple-negative disease. As you may have heard, breast cancer is not one cancer. It's several different kinds of cancer. There's triple-negative disease, HER2-positive disease, estrogen receptor-positive disease. So the first immune therapies were in triple-negative disease, and the reason there was a reason for that, and that is that triple-negative cancers seem like they're the ones who are at least most commonly using this escape mechanism, this PD-L1 expression to get around the immune system. So they have the highest rate of PD-L1 expression, which is again the protein that turns off the immune system. They have the highest rate of mutations, which is part of the reason why triple-negative breast cancers have been difficult to treat, but again, that makes them easier for the immune system to find. And going along with that, if you look at a triple-negative breast cancer under the microscope, usually they're filled with immune cells. So again, the immune system is recognizing those cancers, those triple-negative cancers, but they're not doing anything. They're essentially put to sleep. And the thought was the reason why they're not active is because the cancer is turning them off using this PD-L1 protein. So the first trials, there were 2 of these trials done of these same kind of antibodies against PD-L1 or PD-1. They were done in triple-negative breast cancer, and our group at Dana-Farber participated in one of these. And essentially we got the same kind of results, maybe not quite as impressive, but the same overall feel as those studies that that I showed you in melanoma, which is that some cancers did grow, but a lot of them either shrank or stayed stable, again, for prolonged periods of time. This was an early study, so we only have data out to 1 year, but many of these patients stayed on treatment for several years. So this was one trial, this was another small trial, again, similar kinds of results. some of these cancers at least were essentially the immune system was activated and that effect of the immune system continued to work for a prolonged period of time. So what do these trials tell us? Well, again, they provide proof of concept that immunotherapy can work in at least triple-negative breast cancer. And that was these two trials were the first example of immune therapies working in breast cancer. I should point out, just for transparency, that all the patients on these trials had PD-L1 positive triple-negative breast cancer. So again, we had to know that the cancers made this PD-L1 protein before the patients went on the study, because we thought those are the cancers that were going to work, and not everybody benefited. Only a fraction of the patients showed this real pronounced effect. So clearly there's something there, but we have work to do. So where are we going now? So right now these drugs like Keytruda, these PD-L1 inhibitors, are being tested in huge international trials. We don't know the results of any of them yet. They're also being combined with chemotherapy, because chemotherapy actually also is a way to stimulate the immune system in some aspects. And now there's a number of other immunotherapies that are being tested as well. And that's what a lot of what we're doing in the Breast Oncology Center at Dana-Farber is starting to test other immunotherapies as well. So what about other kinds of breast cancer? So HER2 positive disease is kind of not quite as immunogenic as triple negative cancers. They do have a decent number of mutations, and some of them make PD-L1, but the potentially cool thing about HER2 positive disease is that we already use drugs that have some ability to stimulate the immune system. Many of you have heard of trastuzumab or Herceptin, which was the first targeted drug against HER2. Turns out that not only does it block HER2, but it also stimulates the immune system. So there are data in the laboratory that adding our already used drugs like Herceptin plus immune therapies can work well together, and there will be data likely in the near future showing that that's an effective treatment for HER2 positive disease. We have a trial going on now that are looking at other drugs combined with PD-1 inhibitors in HER2 positive cancers, and we think that's going to be a very encouraging approach. And lastly, hormone receptor positive disease. So these are the most common kinds of breast cancer. And unfortunately, while we have a lot of other drugs that work well in ER positive disease, these cancers tend to not have a lot of mutations, and the immune system seems to have a lot of trouble recognizing these cancers. So what we're doing now, and what many groups are doing, is trying to figure out a way to make these ER positive cancers more what we call immunogenic, more recognizable by the immune system, so that we can start using drugs like PD-1 inhibitors. So I've talked about PD-1. Turns out that there's a lot of other on-off switches on T cells. On the right are all the off switches, and on the left are all the on switches. So it's actually, as usual in medicine, a lot more complicated than we originally thought. But that's good, because we can develop drugs, and drugs are already being developed, that turn on that block some of the off switches and stimulate some of the on switches. And so these are the kind of drugs now that are being entered into clinical trials. So we have proof of concept that immunotherapy can work in cancer, and that's really, again, a really groundbreaking thing. But we're just scratching the surface. And the real hope, and I think that there's a lot of reason to think this is true, that once we start having this huge array of tools at our fingertips to start combining these different kinds of drugs, we're really going to see a lot of progress. And so I think it's an exciting time. We have a lot of work to do, but there's a lot of potential for real benefit for our patients. So a lot of promise in other cancers besides breast cancer. These have completely revolutionized the treatment of melanoma, of non-small cell lung cancer. The data in breast cancer is very early, but these drugs can work in breast cancer, and right now the field is really focused virtually a huge fraction of our new clinical trials are working on how to develop immunotherapies in breast cancer to keep pushing the field forward. So in the next few years, we're going to have a lot more data about this. I'm just, I seem to have lost one slide there, but how can Komen, how can you all help move this along? Again, clearly there's benefit, but we have work to do. And so I think the biggest thing that people can do, and this is the way I end most of my talks, is talk about participation in clinical trials. All the data I showed you is from patients who went on clinical trials. So as those of you who are affiliated with Komen, those of you who know Komen, those of you who are interested in how we advance breast cancer treatment, when people come up to you and say, patients come up to you and ask what to do, encouraging people to participate in clinical trials is incredibly important, because the only way we're going to develop any of these new therapies is by patients participating in clinical trials. And you can see we have a ridiculous number of new drugs that are currently available in clinical testing. So we have to have more robust participation in clinical trials. So I would encourage you to try to get that word out. So I'll stop there. I'm happy to take questions.